A general view of our methodology is confirmed by the double digestion, electrophoresis, alkaline phosphatase, ligation, production, extraction, and infection in prostate cell. Finally, our real-time RT-PCR. To accomplish the site lysine is necessary the implementation of sRNA. There are products of double-stranded RNA by the dicer enzyme which cut the molecule in fragments of 25 to 21 nucleotides, or chemical synthesis. Finally, the combination of single-strand RNA with arginine from RIS complete to join with mRNA. Therefore, the mRNA of the interest gene will go down due to the fact that the sRNA inhibits the translation process. The selected vector is adenovirus V2, that is a PAAV UCV with a length of 5.1 kilobase. We select this vector because to accomplish the transfection and the introduction of the gene of interest into the cell is easier with this vector. To accomplish this process is necessary to find a proper enzymes that will do the double digestion. This method takes advantage to clip the DNA at a specific sequence. Our temperature to work is 37 degrees with a cofactor or Mg2, and our restriction enzymes are EQR1 and HindI3. These restriction sequences are not present in our gene of interest. Once the double digestion is complete, the purification process by agarose electrophoresis removes the small pieces of DNA as a result of the double digestion. To prevent the recirculation of the vector is useful the implementation of shrimp alkaline phosphatase, which removes the phosphate group of the nucleotides. The ligation of the gene of interest and the vector is performed by the T4 ligase, which catalyzes covalent phosphodiester linkage, and the ligase buffer that contains ATP. To make an effective ligation process, it's important to consider too high enough concentration of DNA, the proper ratio between the vector and the gene, which is approximately 1 to 3, to make sure that the ligation will occur properly. Also, the agar electrophoresis and the double digestion that are our previous steps are essential to increase the ligation efficiency.
The production of the recombinant vector is going to happen into human embryonic kidney cells. The first step for this procedure is to thaw the hex cells in a water bath at 37 degrees. Then we need to seed the cells, and the conditions to incubate is 37 degrees, the 95% of humidity, and 5% of CO2. It is necessary to prepare protein and genetic material mix at a concentration ratio of 1/3.5. Then we take 6 cell culture dishes out of the incubator and completely aspirate the medium from each culture dish in a laminar flow hood. We add 12 milliliters of DMEM1 solution to each dish and mix the PEI and genetic material and add 2 milliliters of that mix to each culture dish. Then we need to incubate again the cells for 5 hours at 37 degrees with the same conditions of humidity and the 5% of CO2. We add an additional 12 milliliters of DMEM10 solution to each culture dish, and we incubate the cells up to 72 hours at 37 degrees with the same conditions, and we use a cell scraper to carefully detach the cell from the culture dish. We collect the medium and the cells in a 50 milliliters conical tube and centrifuge the conical tubes. Carefully discard the supernatant from each tube and place the cell pellets back on the ice. Finally, resuspend each cell pellet in 2 milliliters of lysis buffer directly in the 50 milliliters conical tube.
A Beckman Avance J301 ultracentrifuge and a JS 24.15 roller are used. The roller is previously cooled to 4 degrees Celsius. 15 milliliters Bialumber tubes are used. Then 4 milliliters of cesium chloride with a density of 1.4 are added. Then it's carefully covered with 3 milliliters of cesium chloride of density 1.2. In this way, the discontinuous gradient of cesium chloride is formed. On the discontinuous gradient, 5 to 8 milliliters of vital sample is added. Then it's centrifuged at 100,000 Sg for 90 minutes at 4 degrees Celsius. The result of the centrifugation is a gradient, in which in the upper part are the cellular debris, then the cesium chloride of density 1.2, then a band of empty capsid virus is observed, and another band with a density of 1.34 that corresponds to the integral viral particles, and finally the cesium chloride of density 1.4 is found. To conclude, the impurities will be aspirated from the top of the gradient. Then, with a P1000 micropipette, the viral band is aspirated in approximately 1 ml and transferred to a centricon of 100 kilodalton. Then, 2 washes are carried out with 10 mmol 3 hydrochloric acid pH 8 and 2 mmol magnesium chloride, and then centrifuge at 600,000 Hg with a Sorval RC28S centrifuge and SM24 roller at 4 degrees Celsius. Each wash lasts 2 hours. Finally, the viral concentrate is resuspended in the viral storage solution.
Transfection is a process in which genetic material is introduced into a culture cell, the end result of which is transgenesis. The methods of transfection is a biological, physical, and chemical. The microinjection is a process of transfection of physical method, in which it is sought to inject mainly DNA to the nucleus of a cell by means of microneedles. The injections are made with a microneedles with an external diameter of 1.0 mm and the internal diameter of 0.78 mm. The cells can be injected under pressure using a microinjector and micromanipulator. A microinjection microscope is used to visualize the cells. The recommended injection pressure is 79 mm of mercury, and the recommended compression pressure is 50 mm of mercury.
To prove the modifications in the messenger RNA, the implementation of the real-time PCR is necessary. First of all, the total RNA is extracted from the interest cells and the messenger RNA fraction is separated. cDNA molecule is synthesized by each messenger RNA molecule to be transformed into double strand using the DNA polymerase.